cells, for whatever reason, have evolved in a situation where we like high levels of potassium and little bits of sodium inside the cell. I wrote this big little to indicate concentration differences. But outside the cell, there's a huge amount of sodium and a little tiny bit of potassium. So what we like is kind of opposite to what is in typical saline solution. So we have in our membrane a little pumping device that grabs what little potassium is there and pulls it into a region where there's already high level of potassium. Is that energetically uphill or downhill? Uphill, why? Because you're going against the concentration gradient. If anything, diffusion would want to get stuff out to equalize it. So to make an even bigger difference means it's energetically uphill. And the same with the sodium. You're pumping sodium out into a high concentration. So that's unfavorable, but that's the way the cell likes to operate, high potassium inside. So we have the sodium-potassium ATPase, it's called the sodium-potassium pump. It's got kind of odd stoichiometry. It pumps out simultaneously 3 sodiums going out, 2 potassiums coming in, and none of that happens without the hydrolysis of 1 ATP. So 3 sodiums go out, 2 potassiums come in, an ATP gets hydrolyzed on the inside. All of our cells have this. And it only works as a pump because of the ATP hydrolysis. Another example, of course, is muscles. You have the biology classes deal with this kind of structure. You know that muscles are these fibers that slide next to each other. You've got these thin actin molecules, thicker myosin molecules that are linked. There's little extensions coming off the myosins that contact the actin. And during muscle contraction, all that's happening is these guys are sliding past each other, shortening up the sarcomere. At a molecular level, what's happening is that these little extensions, these so-called headpieces on the myosin, are contacting the actin, in some cases then breaking free and bending over and reattaching a little bit further along. It's like a millipede walking with 100 little feet. It kind of walks along the actin molecules. That walking happens due to ATP hydrolysis. So here's the headpiece attached to the thinner actin molecule. When ATP binds to it, it breaks the link between the myosin and the actin. And when this is an ATP's activity, when it hydrolyzes the ATP, it cocks the headpiece back so it can grab it in a new position. So the energy of the ATP is used to change the conformation of the protein. It reattaches when the phosphate falls off, and then when the ADP product falls off, you have the power stroke, where it goes back to the starting conformation. So muscle contraction is really a protein, a small part of a protein, doing this, driven by ATP.